Hello everyone, Mr. Linder here. Let's talk about action potentials in cardiac muscle cells. So in the heart, there are two types of cardiac muscle cells. There are the contractile cardiac muscle cells, and those are the cells that pump blood. So I like to call those like the 99% of cardiac muscle cells in the heart. These are the contractile cardiac muscle cells. And so they're pumping blood to the body and to the lungs. The other type of cardiac muscle cells are the 1% of cardiac muscle cells in the heart, and these are the conduction system cells. And these are going to be the SA node, the AV node, the AV bundle, the bundle branches, the Purkinje fibers. These are the cardiac muscle cells that are sending electrical signals across the heart so that the cardiac contractile cells can respond and pump blood. So since there are essentially two types of cardiac muscle cells within the heart, there's in essence two types of action potentials that we see taking place within the heart. So let's start with the action potential of the cardiac contractile cells. So the cardiac contractile cells, again, are the cells that are going to be pumping blood to the lungs and pumping blood to the body. And so those cardiac muscle cells generate a specific type of action potential. So when you analyze the electrical signaling that's traveling across the plasma membrane of these cells, you get a specific action potential type recording. And the action potential is shown on the screen. And it's similar to what you see with an action potential in a neuron, so in the nervous system, but it actually is different as well. When you look at a neuronal action potential, you'll notice that it's very, very fast. The duration of a neuronal action potential is a few milliseconds of time. If you look at the action potential of a cardiac contractile cell, you notice that the timing is over 200 milliseconds of time. So there's more time associated with this type of action potential. The other thing that you're going to notice that's different than what you see with a neuron is that there's calcium involved in cardiac contractile cell action potentials. And there's also calcium involved in the conduction system action potentials as well. We don't see calcium in neuronal action potentials. In neurons, we focus on sodium and potassium. And we have sodium and potassium involved in the heart as well, but you get that third ion, which is calcium, involved in these action potentials. So what's going on in the contractile cardiac muscle action potentials? So what we see is essentially starting at around negative 90 millivolts. We're not exactly at negative 90 millivolts for resting membrane potential, but it's pretty close. Negative 90 millivolts is actually the equilibrium potential for potassium. So we're close to negative 90 millivolts, but we're not going to be exactly negative 90 because that would be an equilibrium potential for potassium. But we have a resting membrane potential of about negative 90 millivolts. When there is a depolarization wave that comes from the conduction system of the heart, so as electrical activity is spreading throughout the heart, then we're going to see electrical signals essentially moving through intercalated discs, so moving through gap junctions, and that's going to affect the resting membrane potential of the cardiac contractile cells. And so as the resting membrane potential begins to change, we begin to see a depolarization to threshold. So we're seeing a change from resting membrane potential. A depolarization is going in a more positive direction. And so we're starting to move towards threshold for the sodium voltage gated channels that are in the plasma membrane of the cardiac contractile cells. So when you get to threshold, we're going to see an opening. So we're going to open sodium voltage gated channels. When you open sodium voltage gated channels along the plasma membrane, That's going to allow a greater influx of sodium. So we see an increase in the permeability of sodium. And so as sodium enters into these cardiac contractile cells, we get a much steeper depolarization that takes place. As the sodium voltage gated channels close, there's going to be a decreased permeability of sodium. There is some potassium that is going to enter into the cardiac contractile cells. That's going to start a slight repolarization event, but that's actually going to be counteracted by a change in calcium permeability. So as the cell begins to repolarize, you're going to see calcium voltage gated channels open. And that's the big difference that is different from a neuron. So calcium voltage gated channels opening is actually going to allow calcium to enter into the contractile cells, and that's going to create this plateau that we see here that is preventing the contractile cells from returning to resting membrane potential. And so what we get here is this elongated action potential. So calcium voltage gated channels opening, allowing calcium, so there's a calcium influx into the cells, which is essentially maintaining this depolarized state. Calcium voltage gated channels, though, are eventually going to close. And so we're going to see a decrease in calcium permeability, and then we're going to see an increase in potassium permeability. And that's because we see the opening of the potassium voltage gated channels. As potassium voltage gated channels open, and there's an efflux of potassium, so potassium is going to go out of these contractile cells, then we're going to start repolarizing in greater magnitude back towards resting membrane potential for the cardiac contractile cells. So what does this all mean for the cardiac contractile cells? Because of that calcium influx that is maintaining that plateau, you're seeing this elongated action potential. So we have action potentials that are lasting for over 200 milliseconds of time. Now, this is important because it is extending the refractory period for these cardiac contractile cells. So this is cardiac muscle compared to skeletal muscle. In skeletal muscle, we see a very small refractory period. So you can actually stimulate skeletal muscle over and over and over again, which will actually allow for a summation of contractile events and will lead to what we call tetanus in skeletal muscle. And tetanus is that steady, sustained muscle contraction. In contrast, for cardiac muscle, because there's a long refractory period, you're not going to have these contractions that are allowed to summate. So what we see is muscle contraction followed by muscle relaxation. You can see that in this diagram, muscle contraction followed by muscle relaxation. This is the tension that you see in the cardiac muscle, but you'll notice that the tension drops before you see another contraction followed by another state of relaxation. And that's because of these longer refractory periods. So these action potentials that last for 200 to 250 milliseconds compared to, say, 5 milliseconds of time, is allowing for cardiac muscle to stay out of tetanus. So no tetanus in cardiac muscle. Under normal circumstances, normal functioning physiology for cardiac muscle cells, there's no tetanus. And that allows the heart to not be in a state of steady contraction. Instead, you have contraction followed by relaxation. That relaxation, right, that relaxation that we see in cardiac muscle is vitally important because this allows for the filling of the heart. Okay. So as cardiac muscle is allowed to relax, as the heart is allowed to relax, the pressure within the heart can drop, and then blood can return to the heart so that then you can pump that blood out to the lungs and out to the body. So you need a refractory period in cardiac muscle in order for the heart to work and function effectively. That is why tachycardia is so dangerous for the heart. When the heart doesn't have time to relax, when there's no relaxation of the heart, then you can't have proper filling of the heart. So in order to prevent that, we have these action potentials in these contractile cells that have very long refractory periods, and that's because of that calcium voltage gated channel that plateaus and keeps that depolarized state. The other type of action potential that you see in the heart is the action potential associated with the conduction system, or what we call the autorhythmic cells of the heart. So the autorhythmic cells of the heart, or the pacemaker cells of the heart, have a different type of action potential. What you'll notice with the conduction system, so let's say we're talking about the SA node. So our SA node has our pacemaker cells, our autorhythmic cells of the heart. They actually have an unstable resting membrane potential. So resting membrane potential for these types of cells, these 1% of cells, is typically around about negative 60 millivolts. So on this diagram, we're looking at about negative 60 millivolts as being our resting membrane potential. However, it's unstable. It doesn't stay at negative 60 millivolts. In fact, it slowly moves towards threshold on its own. That's why these are autorhythmic cells. They don't need the nervous system in order to get to threshold and create action potentials. This unstable resting membrane potential and this slow drift to threshold is known as the pacemaker potential. So there's this slow movement towards threshold known as a pacemaker potential, and it's because of the types of channels that we see within the SA node cells and other obviously cells of the conduction system. So if we were to look at an SA node cell, what you would actually see is specific types of channels known as HCN channels. The HCN channels stands for hyperpolarizing cyclic nucleotide channels. These are specific types of channels that basically are open at resting membrane potential, this negative 60 millivolts, and they are allowing sodium and potassium to move through the channel. So we have an influx of sodium and an efflux of potassium through these hyperpolarizing cyclic nucleotide channels. They're affected by the hyperpolarization that happens when you repolarize cells. They're also affected by cyclic nucleotides, which would be cyclic AMP. And I'll talk more about that in just a minute. So these HCN channels that are found in the SA node cell are allowing sodium and potassium to move through it. There's a greater movement of sodium, so there's a net sodium influx. There's a greater movement of sodium into the cell than potassium going out, which is going to cause the cell to slowly depolarize. And the reason there's more sodium coming in is because there's a greater sodium electrochemical driving force. I'll just put EC driving force. The electrochemical driving force, so the diffusion of sodium into the cell and the electrical attraction of sodium into the cell is greater than that of potassium. So we could write something like the sodium electrochemical driving force is greater than that of potassium. Okay, so electrochemical and electrochemical. So the electrochemical driving force is greater for sodium than it is for potassium. So even though both ions move through this channel, there's more sodium coming in than potassium going out. And that's what's causing this pacemaker potential. That's what's leading us to threshold in the SA node in these pacemaker autorhythmic cells. And so that's why you don't need influence from the central nervous system for your heart to contract. You can use these specialized types of HCN channels. So more sodium coming in than potassium going out is leading us to threshold. There is some movement of calcium coming in as well. So there's calcium entry from the extracellular fluid that is also helping us to get to threshold. And so when you reach threshold, what happens? So at about negative 40 millivolts, that is the threshold for these autorhythmic cells. So let's say the SA node cells. Once you hit threshold, you're going to open calcium voltage gated channels. Now that's different, again, than what you see in neurons. In neurons, you would be opening sodium voltage-gated channels, but this is cardiac action potential. So in this case, when you hit threshold, you open calcium voltage-gated channels. That greater influx of calcium is what's giving us the depolarization spike that you see in these autorhythmic action potentials. Calcium voltage-gated channels open at threshold, influx of calcium, depolarization spike. Those calcium channels are going to close, and then we're going to see the opening of potassium voltage-gated channels. That's actually the same thing that you see happening in neurons. And so the opening of potassium voltage-gated channels is going to allow for potassium to flow out of the conductive system cells. And so that's going to repolarize the cell, repolarize the membrane, and bring us back to that resting membrane potential state. Again, it's an unstable, though, resting membrane potential. So as soon as you essentially repolarize and have a slight hyperpolarization, these are, again, hyperpolarizing cyclic nucleotide channels. So they are open again. And so that influx of sodium that is greater than the efflux of potassium is allowing us to start that pacemaker potential again, bring us to threshold, open calcium voltage gated channels, and we're again sending an electrical signal across the heart so that the heart can beat again. The last thing to note, though, about this process is that this particular action potential can be influenced by the nervous system. So although the conductive system cells generate their own action potentials without the nervous system, we do know that the autonomic nervous system can influence heart rate. So the autonomic nervous system can change heart rate. And we know that because the parasympathetic and sympathetic branches of the autonomic nervous system change our heart rate. So the parasympathetic decreases heart rate, and the sympathetic nervous system can increase heart rate. And so the question becomes, how does the autonomic nervous system influence these action potentials? So this is the normal action potential rate. So seen in gray, this is the normal rate. Okay. But notice under the influence of the parasympathetic nervous system, here we have one signal, and then we have a second signal, which is much more spread out. So the normal here is 3 essentially heartbeats. And now under the influence of the parasympathetic, we have 2 heartbeats. Okay, so what is slowing down the heart rate? So let's take a look at what would be happening in an SA node cell under the influence of the parasympathetic nervous system. So the parasympathetic nervous system releases a neurotransmitter known as acetylcholine. And acetylcholine can bind to receptors in the SA node cells. So these receptors are going to be muscarinic 2 receptors. And so when acetylcholine binds to the muscarinic 2 receptor, it activates a G protein system. So G proteins are made of 3 subunits, alpha, beta, and gamma. And so when acetylcholine binds to a muscarinic 2 receptor, what it is doing, it is activating the G protein. And so basically when a G protein becomes active, it splits the alpha from the beta gamma subunit. In this scenario, the beta gamma subunit can break away from the alpha subunit, and it can move through the plasma membrane, and it can actually affect potassium channels. So this is a potassium channel. Now, you have to remember that the HCN channels are already active. And when you get back to that hyperpolarized state, these HCN channels are active. They're open again. So we know sodium's coming in, and we know potassium's going out. But we know that the sodium going in is greater than the potassium going out. So if you want to slow down what this cell is doing, you can affect potassium channels. And what that's going to do is that's going to allow for more potassium to flow from this cell. So we have potassium, okay, moving out of these potassium channels, and that's increasing the potassium efflux. So what's happening is, as the sodium is coming in, and there's a small amount of potassium going out normally, but now there's a greater amount of potassium going out because of the influence of acetylcholine, right? So acetylcholine from the parasympathetic nervous system is activating these muscarinic 2 receptors, and it's increasing the amount of potassium going out of the cell that normally would be going out. You're seeing that it's taking longer for the pacemaker potential to get you to threshold. This is the normal pacemaker potential reaching threshold. And now we have the influence of acetylcholine making it take longer to get to threshold because of the greater efflux of potassium from these SA node cells. And so if it takes longer to get to threshold, you're going to have fewer action potential signals. Okay? So if it takes longer to reach threshold, then you have less action potential signals. And so if there's less action potential signals, then you're going to have a decreased heart rate or a slower heart rate. What's going on, though, to increase heart rate? Well, to increase heart rate, again, notice over here the normal is in gray. So this is, again, 3 heartbeats in this amount of time. But here, if we're under the influence of sympathetic, notice we have 1 heartbeat, 2, 3, 4, 5 in the same duration of time. So the heart rate is getting faster. So how is it that we can influence the SA node cells and make these cells send action potentials at a greater rate? So what is taking place in order for these cells to go faster? So here we have the nervous system. And so this is the sympathetic part of the autonomic nervous system. And in this case, we can release norepinephrine. And norepinephrine is going to have a stimulatory effect on the SA node cells. And in order to do that, it has to bind to a receptor. This is what we call a beta-1 receptor of the heart. And the beta-1 receptor that is influenced by norepinephrine also activates a G protein system. So again, we have the alpha, beta, gamma scenario. So 3 subunits to the G protein. When norepinephrine activates a beta-1, the active subunit is the alpha. So the alpha becomes the stimulatory. Sometimes they'll put a little S connected to that. So this is the alpha stimulatory subunit. And what that's going to do is that's going to activate an enzyme called adenylate cyclase. Adenylate cyclase becomes active in the plasma membrane of the SA node cell. And adenylate cyclase is going to convert ATP into cyclic AMP. So again, this is happening inside of the cell. So cyclic AMP is produced, and that is a cyclic nucleotide. And it turns out that cyclic AMP can affect the HCN channels because the HCN channels are hyperpolarizing cyclic nucleotide channels. So norepinephrine binds to a beta-1 receptor, activates the alpha subunit, becomes active, turns on adenylate cyclase, so you create cyclic AMP. Cyclic AMP is going to influence the HCN channels. It's going to open more HCN channels, and so you're going to have a greater influx of sodium through these HCN channels. More sodium coming into the SA node cell is going to make the pacemaker potential depolarize faster, and so you're able to get to threshold faster. And so if you can get to threshold faster, then you can have more action potential signals, and therefore you can have a faster heart rate. And so the autonomic nervous system changes heart rate by either slowing down the pacemaker potential or speeding up the pacemaker potential. So the autorhythmic cells can send signals across the heart without the influence of the nervous system, but you can change the pacemaker potential through the autonomic nervous system by simply using acetylcholine to decrease heart rate or use norepinephrine to increase heart rate. So those are the types of action potentials that we see in the heart. We have the action potential of the cardiac contractile cells, and then we have the action potentials of the autorhythmic cells. I hope that helps. Take care.